All right, friends, welcome back. We're going to continue our discussion of nucleotides and nucleobases and so on. Last time I introduced you to the purines and the pyrimidines. These are the heterocyclic bases that end up being bonded to the ribose ring. The purines have this fused 5- and 6-membered ring. They both share the same core structure of carbon and nitrogen sigma bonds, but vary in terms of what functional groups they have on this side of the molecule. And I've highlighted the different functional groups in red and blue respectively, depending on whether it can donate a hydrogen bond or accept a hydrogen bond. Now, I always make mistakes about whether something is a hydrogen bond donor versus acceptor because, as an organic chemist, I'm used to thinking about nucleophiles and electrophiles. Nucleophiles are electron donors, and electrophiles are electron acceptors. So you can see the sort of confusion. I always look at what I've called here a hydrogen bond acceptor and think, oh, lone pair, nucleophile, therefore donor, and I get it wrong basically every time. So I'm going to apologize in advance for doing that. Understand that a hydrogen bond donor is something that has an electron-poor proton because the proton's bonded to some electronegative heteroatom, whereas a hydrogen bond acceptor is an electronegative heteroatom with a lone pair that can interact with an electron-poor hydrogen. For adenine, you've got a hydrogen bond donor here in this exocyclic amino group, and then you've got hydrogen bond acceptor here. You have two other hydrogen bond acceptors, but these two are the ones that are involved in Watson-Crick hydrogen bonding. In guanine, though, you have a hydrogen bond acceptor and then two hydrogen bond donors. So guanine has more options for hydrogen bonding than adenine does. Over here are the pyrimidine rings. These are the 6-membered rings. Thymine and uracil are the same over here. Uracil lacks this methyl group. And thymine has this pattern of hydrogen bond acceptor, then donor, and it has another acceptor here, though we will see that adenine lacks a complementary group for thymine at the corresponding position. So it's just these two that are going to participate in Watson-Crick hydrogen bonding for thymine. In contrast, cytosine, the other pyrimidine, has 3 groups that can participate in Watson-Crick hydrogen bonding. The exocyclic amino group here is a hydrogen bond donor, and then you have 2 acceptors. And notice that the pattern of donor, acceptor, acceptor is complementary on cytosine to the pattern of acceptor, donor, donor on guanine. They're complementary to each other. Okay, questions so far about those structures? Yeah?
I'm guessing because there are 3 groups that can connect with G and guanine and cytosine, that would be a stronger interaction between those two.
Right, so the question is the fact that G and C can form more hydrogen bonds than A and T mean that a GC pair is more stable than an AT pair? And the answer is yes in the DNA duplex. A DNA, double-stranded DNA that has more GC pairs will be more stable than one that has fewer GC pairs. That has some consequences for the PCR reaction, though I think we won't actually get to that in this class. So, all right, others? So let's talk about the hydrogen bonding in what we're going to call a Watson-Crick base pair. And this is so named for the scientists Francis Crick and James Watson, who were the authors of a 1953 paper in Nature that revealed the double-stranded duplex structure of DNA with base pairs. It turns out their coworker actually did and interpreted the crystallography, though she was not on the paper and did not receive the Nobel Prize, and that sort of ticks me off in retrospect. And so if you are ever on a scientific paper, don't be the jerk who says, oh, well, your contribution was not significant enough, and therefore you should not be an author in order to preserve glory for yourself. That's really bad form, so don't do that, okay? But anyway, end tirade. I'm going to show you here Watson-Crick hydrogen bonding in a CG base pair. What I'm going to show you here would be the same as in a GC base pair. It just depends on which one we're drawing on the left versus the right. So here is the cytosine base. Here's the nitrogen that's attached to the sugar via a glycosidic linkage. And then in cytosine, we have an exocyclic amino group here. We have a ring nitrogen that's nucleophilic there. And then we have a carbonyl oxygen. Oh, shoot, that's an eraser. Carbonyl oxygen that is has lone pairs there. And these can hydrogen bond with complementary groups on guanine. So, nope, that's the wrong one. So in guanine, this is the nitrogen that's bonded to the sugar. You've got an exocyclic amino group here, and then you have a carbonyl oxygen there. And I think that's right on all of the double bonds. So you've got G and C, and you have this complementary lining up of donors here and here with acceptors there and there. And one thing you could ask is, how do you know things are hydrogen bound to each other? And the answer is you sort of guess based on the available information, because it turns out that in crystallography, this is where you can actually grow microscopic crystals of DNA, and then mount them on a crystal holder and then shoot X-rays at them. And as you shoot X-rays through the crystal, and often they're just little tiny things like this, you were probably thinking like, ooh, Kyber crystal, it's kind of awesome, right? No, it's not that cool, but you shoot X-rays through the crystal, and then the crystal diffracts the X-rays, bends them, the pattern of atoms in the crystal bends the X-rays, and they impinge upon a detector. Most of the X-rays go through undiffracted, but then there's often a pattern of spots of diffracted X-rays that you get on the detector, and then you read that pattern, you move the crystal around to get it from all directions, you collect all of the data, and then you analyze it, and you can back calculate out what the atoms look like in the crystal from this diffraction pattern. That involves a ton of math. Computers do it for us now. I think as you may have learned about DNA previously, I think they probably showed you some diffraction pattern from the paper, and it probably looked something like this. And you were supposed to somehow infer from that the DNA was double-stranded. And it's okay if you looked at that and said, I don't know what the heck that means, or how just looking at that could tell you that it's double-stranded DNA. It doesn't. It's just there's a lot of maths that you do to back calculate it all out. All of this is done by computer. Back in the day, they had to do the math and then build a 3D model based on coordinates to try to figure out. This is unassisted by 3D printing. They were literally getting out their rulers and measuring bits of wire and positioning them here and saying, okay, the atom is there, and then making a little ball and sticking it there and so on. It took forever, which is why it was such an important paper when they did this. Anyway, so how do you know things are hydrogen bound with each other? Well, you don't see protons really in crystal structures of large biomolecules. They're just too small to see, or there's too much uncertainty in the data. So what you do is you look for the distance between two heteroatoms that you think are hydrogen bound to each other, and you measure that distance, and in this case it turns out to be anywhere from 2.8, that doesn't look like it, 2.8, around from 2.8 to 3 angstroms. Do we have to memorize that? No, you don't. But that distance, if you were to add up the van der Waals radius or the radius of these atoms, oxygen, hydrogen, and nitrogen, and just add them together, the distance between them that we see in the crystal structure is smaller than that, meaning that these atoms aren't just next to each other, there's actually a bonding type interaction going on. So that's how you can often tell that there's a hydrogen bonding interaction. The other interesting thing is that the distance between the sugar portion of the duplex is around 10.6 angstroms. And that's the kind of information that you can get from the crystal structure. That's going to be important in just a second. But this is what the base pair looks like. And we could draw a similar one with G on this side and C on that side. Okay? Questions you want to ask? So we can draw an AT1 as well. Oops. So to do that, we're going to keep the same basic structure of the rings. I'm just choosing to draw the pyrimidine on the left. So here's the 6-membered ring. Here's the nitrogen involved in the glycosidic linkage. Here's the other nitrogen, and this is thymine. So there's a methyl group here. In uracil, that methyl group wouldn't be there. All right, and then over here you have the guanine ring. Here's the nitrogen involved in the glycosidic linkage with the sugar. Here's the exocyclic amino group that can hydrogen bond with the carbonyl from thymine. Here's the ring nitrogen that can hydrogen bond there. And then there is no group here that is complementary to this oxygen that's sort of just hanging out there. So you have complementary hydrogen bonding between an AT pair. And both of these are called Watson-Crick hydrogen bonds. Notice that the GC pair has 3, whereas the AT pair only has 2. Now, we're jumping ahead a little bit because I haven't said what the sugar phosphate backbone looks like, but all of you have seen a DNA double helix before, presumably, and you've all heard about a major versus minor groove-ish, sort of. So the backbones are here, and then the major groove and minor groove are on the top versus the bottom faces, and I cannot remember which one is major versus minor. But that being said, you can see that there are other groups that are not involved in Watson-Crick hydrogen bonding that nevertheless are pointing outward into either of the grooves. And for example, this other hydrogen could be involved in some other hydrogen bonding partner there. This nitrogen could. The other lone pair on the oxygen here could. The other hydrogen on that exocyclic amino group could, and so on. Transcription factors that bind DNA and promote transcription of the DNA into RNA often recognize specific sequences of DNA, and one of the ways they do that is by having the transcription factors are proteins and they have side chains on them that can hydrogen bond with the specific groups that are in the major and minor groove, right? So you could imagine having a protein positioned here so as to have a hydrogen bond donor that could bond here, and then a hydrogen bond acceptor that could interact there. And so this is how, if you ever are interested, you can go look up in the PDB the structure of transcription factors bound to DNA, and you'll see some of this kind of thing happening. All right. Now at this point, during the 9 o'clock class, somebody asked a question which I thought was interesting, but I promised that I wouldn't put on the test. And then I thought, well, if it's not going to be on the test, we shouldn't talk about it. And then I thought, you guys actually love it when I talk about things that are not going to be on the test, because then you can relax and it limits your exposure to new material. So what the heck? There's an alternative to Watson-Crick hydrogen bonding that's important in the genetic code, and it's called, this alternative hydrogen bonding scheme is called Hoogsteen hydrogen bonding. And it's important in the concept of wobble, which is related to the degeneracy of the genetic code. If none of that sounds familiar, that's okay. You're going to get it in future classes. You may have had some of it already. The concept of wobble is related to the fact that in the ribosome, when a tRNA recognizes, when the anticodon on the tRNA recognizes the codon on the mRNA, there is some flexibility in the third base pair of that codon anticodon recognition. It doesn't have to be exact. And one of the forms of hydrogen bonding that is permitted is Hoogsteen hydrogen bonding. So let me show you what this looks like. And we're going to do this in the context of RNA. So on one side, let's say we have, I'm going to get this wrong, an mRNA coding strand. And then over here, let's say we have the tRNA anticodon, the thing that recognizes the coding strand. And let's say on this side we have uracil. Uracil is just like thymine, only we've lost the methyl group. Now, uracil would normally recognize, uracil would normally recognize adenine, but we can have uracil hydrogen bond with guanine sort of as follows. Sure. Yuck. So here you have the NH group of uracil interacting with the carbonyl oxygen of guanine, and then this ring NH group interacting with the carbonyl oxygen here on uracil. Notice that the geometry there is different than the geometry in Watson-Crick base pairs in DNA. In the TA pair, it's the oxygen on top, and then this NH group that does the hydrogen bonding, whereas in this Hoogsteen pair, it's the other carbonyl oxygen that's involved. Also notice that whereas in Watson-Crick hydrogen bonding, all three of these donor/acceptor groups on guanine are involved. In the Hoogsteen base pair, only two are involved. And this is G. The other consequence of this alternative hydrogen bonding pattern is that the sugar phosphate backbones are not the same distance from each other as they are in a Watson-Crick base pair. So what I'm showing you here would not work well in a DNA duplex, but we're not in a DNA duplex. We've got an anticodon recognizing an mRNA. Okay, so if G can recognize U as well as C, what kind of consequences might that have? Well, let's think about if we have a codon in mRNA, and we're going to write this from 5' to 3'. We haven't really talked about what that means yet, but you might know. And let's suppose the codon is UUU. The appropriate anticodon in the tRNA would be AAA. And by the way, recognition is anti-parallel. We'll get to that maybe at some point. But what we're saying here is that there would be an alternative in which the following anticodon on the tRNA could recognize that UUU, and that would be AAG. Another thing that would mean is that alternatively, the codon UUC would also be recognized by the tRNA. Wait. Could also be recognized by the tRNA AAG. So what that means is that in some cases, the same tRNA can recognize a couple of different codons. Shoot, what am I thinking about here? I'm sorry, I'm getting mixed up just a little bit. Let's just say UUC, and then the anticodon would be GAA. Okay, so what we're saying is that the same tRNA can recognize two different sequences. Okay, that could be fine, provided that these two codons don't actually code for two different amino acids, because then you're in trouble, because then the DNA sequence and the mRNA sequence actually doesn't specify a specific amino acid. So this kind of thing is actually built into the genetic code. Each codon can have, each position in a codon, you've got 3 of them, X, Y, and Z. There's 4 options at each. So that's 64 different options, but only 20 amino acids to code for. And we're going to use 3 of those as stop codons. So we have 61 codons to distribute among the 20 amino acids, which means we're going to double up. So as an example, for the codon that I just introduced, this UUU codon, that codes for phenylalanine. But guess what? To avoid the problem with wobble, the UUC codon also codes for phenylalanine. Right? So genetically, the machinery that synthesizes proteins from RNA has already accommodated this alternative hydrogen bonding pattern that can occur at the 3rd position of the codon. Now, you'll get that at some point. And you may read the word Hoogsteen base pairs in your biochem class, and then you can say, yes, I remember hearing about that once. And then you can go back to my YouTube channel and start watching old videos again and reminisce on old times, okay? All right, so we'll just be done with that. Now, we need to talk a little bit about nucleosides, nucleotides, and so on. And the nomenclature here is just a little bit weird. So recall that we've named the nucleobases adenine, guanine, thymine, and then a weird one, well, two weird ones, uracil and cytosine. Okay, so cytosine seems a little bit different because you got the osine there instead of just the ene. It's going to get worse. So let's talk about what a nucleoside is. A nucleoside is an N-glycoside. Recall that a glycoside is where you have a sugar, and then instead of having a hemiacetal at the anomeric carbon, you have an alcohol at the anomeric carbon such that you now have an acetal. If that were, instead of being an OR group, an NHR group, we would call, or some other kind of nitrogen, we would call that an N-glycoside. So in nucleosides, however you want to say them, the sugar that the base is attached to is a form of ribose. So right now, I'm going to draw for you the ribose sugar. And in a nucleoside, the anomeric carbon, carbon 1, we'll call it 1 prime for reasons that I'll mention in just a little bit. Carbon 1 prime is bonded to one of the nitrogens on the nucleobase, their heterocyclic base, and then I'm going to draw the structure of adenine here. Okay. So you got two portions of this molecule. You've got the nucleobase portion, and each of the atoms in the nucleobase is numbered, but you don't have to worry right now about the numbering scheme. Right now or ever, really, it may come up in biochem. And then here in yellow is the sugar portion, and this is ribose, and atoms of the ribose ring are numbered 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So ribose is a 5-carbon sugar. It forms a 5-membered ring. It's an example of a furanose. When you put ribose and adenine together, it forms, like we've said, a N-glycoside, which we're going to call a nucleoside. And the name of that is going to be adenosine. So if you hear people talk about adenosine, what they mean is adenine attached to ribose, right? Now, there are also deoxy nucleosides, and they differ from regular nucleosides in that the ribose ring is missing an OH group. So if we remove the 2' OH group, we now call this deoxyribose, and we would call the nucleoside deoxyadenosine. And the deoxy reflects the fact that there is no OH group on the 2' carbon. By the way, this deoxy, that's the D from DNA. Whereas the R in RNA comes from the fact that you have the OH group there, so you're dealing with ribose. Question so far? Yeah.
What are the other names for like when guanine is?
Oh, sure. So when, yeah, this is the weird part. If the base is G, the nucleoside is called guanosine. If the base is T, it's called thymidine. And if it's C, it's called cytidine. And if it's uracil, it's called uridine. So guanosine's easy because it's like adenosine, but the purines are the only ones with this ocene name in them. The rest of them, the pyrimidines, we just do weird stuff. Like, and this is what is weird. The C base is called cytosine, which makes it sound like it's a nucleoside because of adenosine and guanosine, but it's not. If it's bonded to the ribose, it's called cytidine, pyrimidine, and so on. That's kind of dumb, but I didn't make the rules, and that's just what people call it. Okay, other questions? Yeah.
So the one with ribose is called adenosine.
The one with adenine attached to ribose is adenosine.
And what's the difference with the other one?
If you replace the nucleobase with one of these, yeah. Okay, others? So that's nucleosides. Now, where do nucleotides come from? Because DNA is deoxyribonucleic acid, but you've heard of nucleotides, and what that means is we call things nucleotides that have phosphates attached to the ribose. So if you take adenosine as an example, and convert the OH group here from being just an OH group to having it involved in a phosphoester, that's now a nucleotide. So this would be, the presence of the phosphate makes it a nucleotide. And we would call this adenosine monophosphate. And some people would put a 5' here to indicate that the phosphate is on the 5' oxygen. Or AMP. And if we lost the OH group on the 2' hydroxyl, the only difference is going to be, we would call this deoxy adenosine 5' monophosphate, or DAMP. Okay? Now there's other forms of AMP. If you take biochemistry, you may learn about CAMP, which is cyclic AMP, and that involves the phosphate here actually bridging these two oxygens. Kind of weird, but that's sort of what we're looking at. Questions? Okay, and now that you have one phosphate group there, you can start to see how you could add additional ones. So let's take, adenosine monophosphate, and let's add another phosphate group to it. Here's the phosphoester linkage. And then here is another linkage, though that linkage is not a phosphoester. In fact, that's a phosphoanhydride. And just like regular esters are more stable than regular anhydrides, so phosphoesters are more stable than phosphoanhydrides. And we would call this adenosine diphosphate, or ADP. Okay, and then you can see where we're going. If we tack an additional phosphate group on there, and I realize I'm copying and pasting the notes will be posted, so don't worry about scribbling down everything. Having 3 phosphates makes this ATP. And in ATP, you have 2 phosphoanhydride linkages. Sorry, ATP. Now, what I haven't drawn are the other sort of deoxy versions. If it was deoxy adenosine diphosphate, we'd call that DADP or DATP. And all of these are important and around in biology. Okay. What else? Yes?
What is stopping additional phosphate from being added?
What's stopping additional phosphates from being added? I don't know. I would just guess stability of the bonds that you're forming, but I don't know. It's also possible that there's just no need for it. It's a good question. I don't know whether that alternative exists. Others? So people will talk about the 3 phospho groups of ATP in different ways. I can't remember exactly, but I think it's alpha, beta, and gamma. It may be backwards from that. And so if you learn someday that it is, hopefully you'll forgive me for getting it wrong. But I think for our purposes, we can talk about it that way. All right. So let's now talk about how you would synthesize an oligonucleotide. All of these are mononucleotides because you have 1 ribose, 1 base, and 1 set of phosphate groups. But now we want to link multiple riboses together through a phosphorus. And that's going to give us the RNA and the DNA. So let's do, as an example, RNA synthesis. I'm going to show you the chemical steps that occur. I'm not going to show you what the enzyme does. This is an enzyme catalyzed process, and the enzyme that does this is called RNA polymerase. But for now, let's assume we're at the very beginning. So we've got here a ribose ring, and we have an OH group here. So we're going to be drawing a nucleoside, and then hopefully, if it's okay, I'm just going to abbreviate the base as A, and then you can go somewhere else in your notes and figure that out. So here you have a molecule of adenosine, and this is the 3' hydroxyl, and this is the 5' hydroxyl group. And then let's say our next base that we want to add is G. So we want to add the guanosine base next. Okay? So to do that, nature is going to use, instead of just the nucleoside guanosine, nature's actually going to use GTP, or guanosine 3' triphosphate. So here's the G, here's the 2' and the 3' OH groups. And then in guanosine triphosphate, the 5' OH group is involved in a phosphoester. And then that phosphate is involved in a phosphoanhydride, which is involved in another phosphoanhydride. Now, if that looks like a lot of negative charges and makes you uncomfortable, that's fine. It turns out that nature provides cations, and frequently the cation is magnesium, which is a divalent cation. So a single magnesium can sit and interact with 2 oxygen groups on the backbone, 2 oxygen groups from the phosphoruses. So I'm going to ignore that, but realize that magnesium is often has to be part of this in order to complement the charges. Okay, so I told you that this linkage is a phosphoanhydride linkage, and that the phosphoanhydride linkage is weak. Another way of saying that is this group here is a good leaving group. And in fact, you'll notice that that is exactly the same leaving group as we've been using in terpene chemistry. That's a pyrophosphate group, right? So what's going to happen is we're first going to remove a proton from the 3' hydroxyl group, and a base will do this in the enzyme active site. Then that oxygen is going to attack the alpha phosphorus, the alpha phosphate group of GTP in sort of SN2 fashion. So let me give us plenty of space to work with. Oops. Nope, don't want to do that. All right. So the negatively charged oxygen, 3' oxygen, is going to attack this phosphorus, and the leaving group will leave. We're going to think about this as being an SN2 reaction. There is some evidence that the mechanism isn't this simple. If you think about the SN2 transition state, it would have a new bond forming here and an old bond breaking there. So a phosphorus sort of attached to 5 different things. There's some evidence that that's actually an intermediate rather than a transition state. And some people will draw this mechanism as sort of like carbonyl chemistry, where you make a bond here and put a negative charge out on this oxygen, and then that negative charge kicks down and the leaving group leaves in a second step. I'm going to keep it simple through this simple SN2 mechanism. Just realize there might be some more details that we're oversimplifying. So when that happens, let's see how best to show. Maybe we'll just show it over here. Now the 3' hydroxyl group of our adenosine is bonded to the phosphate, the alpha phosphate group of what used to be GTP. Okay, and now we have a dinucleotide. We're not going to worry about what it's called. Sometimes people do, but we won't. But notice that instead of having a phosphoanhydride linkage, the new bond we made is a phosphoester. This is the new phosphoester we made. This is the old phosphoester that was present in GTP. This is the alpha phosphorus that was there. We've made a bond between the 3' hydroxyl of one sugar and the 5' hydroxyl of the other. The intermediate group is this phosphodiester. So this now becomes, oops. This then becomes the phosphodiester. And when I say sugar phosphate backbone of DNA or RNA, this is what I'm talking about. Sugar bonded to a base, then phosphate group, then sugar bonded to a base, and so on. And when you hear that DNA and RNA synthesis happens from the 5' direction to the 3' direction, this is what it's talking about. The 3' hydroxyl is the nucleophile. The 5' phosphate group is the electrophile. And just like peptides had directionality, an N-terminus and a C-terminus, so oligonucleotides, that is, a polymer of nucleotides, have directionality. This is the so-called 5' end, and this is the 3' end. And so we will write DNA sequences from 5' to 3'. So in this case, we would go A, and then G, and so on. Okay? All right, questions you want to ask about that structure? Yes?
Are you missing a hydroxy group on your adenosine?
Am I missing a hydroxy group? Yep, thank you for that. Sorry, we just... Yeah, Okay, good. What else? So actually looking at this structure, this is a dinucleotide made of ribose, so it's an RNA nucleotide rather than a DNA. You can see one reason why people will tell you that RNA is not as stable as DNA. And the idea is that in RNA, adjacent to the phosphodiester, you have another OH group. And intramolecular reactions can be fast. So it's possible for this OH group to attack the phosphodiester and kick off the 5' hydroxyl group. And that can happen under even slightly basic conditions. So when you're working with RNA, if you want to keep your RNA molecule intact, you don't expose it to basic conditions, because it can catalyze its own hydrolysis. Another reason why you have to be really careful with RNA is that ribonucleases that chew DNA up are highly active and are highly resistant to denaturation, which means that even if you have tap water that you take to the autoclave and boil it at really high temperature to kill any of the bacteria that are in there, that high temperature will not irreversibly destroy any ribonucleases that are present in that tap water. And then you start using it to dissolve your RNA sample, and the residual ribonucleases that are present in that tap water chew up your RNA, and you just spent hundreds of dollars to reduce an RNA to its component parts. So labs, and ribonucleases are on your skin, they're in your saliva, they're all over the place. And so if you're working in a lab with RNA, usually you have a separate area of the lab that deals with RNA. You have separate pipettes, separate equipment. All of the water you use has to have a ribonuclease inhibitor in it so that your RNA stays intact. So it's a little bit tricky. But it's awesome that we've got mRNA technology, because that was the foundation of the COVID vaccine treatment. Yeah?
You said that we have RNases like everywhere. Does that apply to DNases as well?
The DNA is more stable, and there are nucleases that are for DNA, but they're not as much of a problem because of RNA doesn't generally form a duplex structure or a double-stranded duplex structure, so it's easier to get to, I think. I'm hand-waving at this point. But. Okay, we'll talk again next time, which will not be Friday. Go enjoy your big spring day. Yay! And I'll see you Monday.